This is week 11 and 12, and we're going to cover next generation sequencing and genome sequencing, personalized medicine, and newer molecular techniques. The learning objectives for this week are to explain next generation sequencing and list current and emerging applications, explain whole exome genome sequencing and list current and emerging applications, to use genetics, literature, bioinformatics, and computerized tools in a specific case studies, define and give examples of pharmacogenomics, and to discuss the meaning of personalized medicine and give examples. The sequencing of the genome has brought a set of new approaches to biological investigation, and this includes genomics, bioinformatics, and systems biology. When we look at genomics, it's the study of large sets of genes or gene products, up to and including the entire genome or the entire set of transcripts or proteins. And we see that genomics utilizes the gene chip, in which thousands to hundreds of thousands of DNA sequences can be affixed to spots on a glass chip and used to detect the presence of homologous sequences in a test sample. There's many variations of this technique today, and the cost has decreased dramatically in the past few years. And today, next generation sequencing, which is based on massively parallel sequencing, allows us to sequence individual genomes. Bioinformatics is the use of computers to catalog and analyze large sets of biological data. It helps to identify genes or control elements. It gives us a comparison of sequences among different species, and it can be used to identify functional regions that have been conserved through the process of evolution. Systems biology is an integrated approach to the study of complex biological phenomena. And we find that genes, gene products, proteins, and metabolites interact with each other, and they form these integrated networks. And systems biology involves an effort to understand how the integrated networks are structured. The human genome was sequenced one fragment at a time. It took a long time to do it, as I mentioned before, and it cost a lot of money. Today, there have been a lot of technological advances in DNA sequencing, and so now it's possible to sequence the human genome in a matter of hours to days. Collectively, the process to do this is called next generation sequencing. And if we look at the slide in this diagram here, we can see one approach to massively parallel sequencing, which is part of next generation sequencing. DNA fragments are linked to a common primer sequence. They're then amplified. They're then rendered single-stranded, and they're bound to a solid substrate. After this takes place, the DNA is copied off the templates using blocked bases, each labeled with a different color. The bases are then incorporated one at a time, since the synthesis reaction stops after each base, until the base is chemically unblocked. Each time, the fluorescent signal at each site is photographed, allowing the DNA sequence to be put together for billions of short fragments. The resulting sequence for 3 bases is shown at the bottom here for GTG, CCC, and you can see in the rest here. Please view the 2 accompanying PowerPoints, one on prenatal testing and the other on pediatric exome testing. Pharmacogenomics is a field of research focused on understanding how genes affect individual responses to medications. The long-term goal of pharmacogenomics is to select drugs and dosages best suited for each person. Pharmacogenomics looks at variations in genes for proteins that influence the drug response. In pharmacogenomics, a pharmacogenetic test detects a variant of a single gene that affects drug metabolism, and a pharmacogenomic test detects variants of multiple genes or gene expression patterns that affect drug metabolism. Utilizing genetic testing to guide drug selection offers several advantages. These are identifying patients likely to suffer an adverse reaction to a drug, selecting the drug most likely to be effective, monitoring response to drug treatment, predicting the course of the illness. For example, if we look at the polymorphism of the gene CYP2DG, which encodes for the enzyme debrisquine hydroxylase, it acts on a quarter of all prescription drugs, including codeine, which it converts to the drug's active form, morphine. CYP2DG exists in more than 160 different versions, and the majority do not affect drug responses. Some individuals have hundreds or even thousands of copies of the gene. Typically, people have 2 copies of each gene. Those with extra copies of the gene manufacture an overabundance of the enzyme molecules and metabolize the drug very rapidly. As a result, codeine may be converted to morphine so quickly and completely that a standard dose of the drug can be an overdose. Some variants result in non-functional enzymes, so there is little or no pain relief. If we look at some of the other drugs that are metabolized by this enzyme, analgesics, antiarrhythmics, antidepressants, antihistamines, antipsychotics, beta blockers, cough suppressants. One thing we're looking at with this sort of personalized medicine, and we'll talk about that just in a moment, is that the day may be coming when virtually all treatments are based on genetic information. And that leads us to the term personalized medicine, which NIH has defined as the tailoring of medical treatment to the individual's characteristics of each patient. Today, we've entered an age of personalized medicine, the science of individualized prevention and therapy. And this is a poster showing how personalized medicine is transforming patient care. So we look at personalized medicine and we see that's also referred to as individualized or precision medicine. It's an emerging field that uses the diagnostic tools to identify specific biological markers, often genetic. And we can look and see how it has improved patient outcomes, for example, with cancer and with cystic fibrosis. And some of the things that we're able to do is to diagnose faster. We can target the right medicine and medication to the right patient. We can prevent complications and side effects, and we can improve our outcomes for patients. Now, please go and complete your case study, and this will be the end of your work for week 7. Week 8 is your last week, which contains your test.